Hello, check. Mic testing. Check. Hello. Check. 1, 2, 3. Check. Hello. Check. Hello. Hello. Check.
Silence, please. A very good morning to all of you. It is my honor to welcome you all to the CSIR-CFTRI Platinum Jubilee Lecture Series, celebrating the 75 remarkable years of innovation and research contributions in food and science. Over the decades, CSIR-CFTRI has been the institute at forefront, pioneering the research, setting up the new standards for food standards, and nutrition for the benefit of the society. As part of our 75 celebrations, this lecture series is being featured to distinguished speakers and eminent speakers to contribute and present their related work related to the invaluable contributions of this field. We are glad to the esteemed speaker present today to share his insight and experiences. I cordially welcome the dignitaries, Dr. Rakesh Sharma, the chief guest, the director of Tata Institute Genetics Society, director of CSIR CCMB in 2016 to 2021, and the director, Dr. Sridevi Annapurna Singh, to kindly come over to the dais and occupy the seats. Now, may I kindly request the Director, CSIR-CFTRI, Dr. Sridevi Annapurna Singh, to kindly welcome our chief guest, Dr. Rakesh Mishra, with a flower bouquet. Ma'am. Now, may I request Dr. Giridhar P., Head Department of Food Safety Analytical Quality Control Department, to kindly welcome the directors, Dr. Sridevi Annapurna Singh, with a flower bouquet. Now, I kindly request Dr. Giridhar P. to introduce the chief guest.
Good morning to everyone and welcome. It's my pleasure to introduce our distinguished speaker, Dr. Rakesh Kumar Mishra, a renowned scientist specializing in genomics and epigenetics. Dr. Mishra completed his post-graduation in organic chemistry and obtained a degree from University of Allahabad in 1981, and also his PhD degree he obtained in 1986 from University of Allahabad in nucleic acid chemistry as specialization. Subsequently, he started his professional career as a postdoctoral fellow in September 1986 and during 1988 at IISc Bangalore. Later, he joined in CCMB as a scientist in 1988, and later, so 3 times he had a postdoctoral fellow programs at overseas, like between 1992 and 1995 at the University of Bordeaux, France, and in 1995 and 96 at St. Louis University, USA, and in February 1996 to 1996 again University of Geneva, Switzerland. Again he back to CCMB Hyderabad, and rose to the position of Director CCMB at Hyderabad in 2016 and led that CCMB up to 30th April 2021. During his leadership at CCMB, Professor Mishra has taken initiatives to establish the Atal Incubation Center at CCMB, providing life science entrepreneurs with the infrastructure and the mentorship needed to encourage innovation. CCMB has also played a pivotal role in genomic surveillance of the SARS-CoV-2 virus in India during the COVID-19 pandemic and made major contributions in diagnostics, drug screening, environmental surveillance, and capacity building in various efforts against the pandemic. I think in those days you might have seen that in 2018, in many national dailies also has come in news that under his leadership, CCMB has taken an initiative to develop lab-grown meat. So that is the first time that's what. Subsequently, in 2021, he joined as advisor and JC Bose National Fellow at CCMB itself, and also presently continuing there as a distinguished emeritus professor of ACSIR, that is Academy of Scientific and Innovative Research at CCMB. In 2021 May, he assumed the charge of Director of Tata Institute for Genetics and Society at Bangalore, and currently he is associated as the Director of this institute. Professor Rakesh Mishra, research areas, if you look into that one, it's his expertise in genome organization focusing on evolutionarily conserved regions, structure of a chromatin material, and epigenetic regulation of genes during the embryogenic developmental stages in various experimental models. Some of the focused areas includes antisense targeting protozoan parasites in Leishmania, role of small nucleolar RNAs in ribosomal RNA maturation in Xenopus, chromatin organization and regulation of Hox genes in Drosophila, comparative and functional genomics of non-coding DNA, epigenetic regulation and cellular memory, and homeotic genetic regulation and development. So these are the various focused areas wherein Professor Mishra has excelled. Professor Rakesh Kumar Mishra is an elected fellow of Indian National Science Academy, New Delhi, Indian Academy of Sciences, Bangalore, National Academy of Sciences in Allahabad, and Andhra Pradesh Academy of Sciences, Telangana Academy of Sciences, elected fellow of Guha Research Conference, and JC Bose National Fellow. Professor Mishra has guided to 28 students for their PhD and guided more than 50 students for post-graduate dissertation works. And to his credit, he has more than 185 publications in various reputed international journals and 3 patents. Currently, he is the editorial board member of the journals like Development, Genes and Evolution. Earlier, the same journal is known as Roxas Archives of Developmental Biology. Similarly, the Mechanisms of Development, this is another prestigious journal. This is an official journal of the International Society of Developmental Biology. Gene Expression Patterns, Journal of Proteins and Proteomics, these are the other journals along with the Frontiers in Cell and Developmental Biology, Morphogenesis and Patterning. He served and also serving as the chairman and as a member of various task force committees of CSIR and DBT. More than 26 committees presently. Now, a couple of things I would like to highlight. He is a chairman for the High Power Committee for Review of Recruitment and Assessment Rules of CSIR. He is the chairman and a committee to review the scientific leadership, that is scientist H and positions in CSIR, and a member advisory committee of CSIR Unit for Research and Development of Information Product, that is URDIP, member High Performance Computing Policy Committee of CSIR, and member of a task force on research on technology development, silk and applications in biomaterials, that is the DBT task force committee, and also he is a member of a scientific and technical appraisal and advisory groups of knowledge generation and discovery research, new tools and technologies, again it is the DBT one. Similarly, the chairman of RCGM, that is a review committee on genetic manipulation of DBT, and chairman subcommittee of DNA barcode at Indian Pharmacopoeia Commission, and a member of Niti Ayog's technical advisory committee to assess evaluation system of performance of Government of India. Now, please join me in giving a warm welcome to Dr. Rakesh Kumar Mishra, who will be sharing his insights on genome technologies for One Health, novel therapeutics to nutritional security.
So, first slide changing, yeah, okay. Pointer will work, or? Okay. All right. While this is happening, so first I would like to thank Dr. Sridevi for inviting me and the staff of CFTIR. It's one of the beautiful labs, fabulous lab of CSIR, and I've been here several times, but this time after maybe 4 or 5 years. It's always a pleasure to come to Mysore, particularly when you are in Bangalore, you want to escape and come to Mysore. So I'm very happy to come here very often. And thank you for the very, very elaborate and generous introduction, which anyway, we'll not talk about that. So let's now discuss what I decided to present to all of you, how genome technologies are now going to have impact in our life if we manage them well in a positive way. Otherwise, we will be buying things from others at unusual prices. So, I have put the title with little bit broad in the sense, as you may be knowing, the One Health means the health of the planet. You cannot segregate the health of humans without considering the health of animals and health of plants. And the genomics is going to play a major role. Some of the examples I'm going to show, which will vary from, range from novel therapeutics to nutritional security. So I will just take few minutes to tell you, which all of you must be knowing, that in an organism, this complete set of the DNA that we have is our genome. And this is the complete information set. Okay, it's a little weak, it doesn't matter. Which is the basis of life forms on all the organisms on this planet. More importantly, or equally importantly, it is also the substrate for evolution. So changes that happen, then they will be continued for the generation to generation. So that is why it's so important. What has happened in last 30 years is that we have got ability now to read it, the complete genome we can read, and we can even edit it. We can go and make precise changes in the genome, which was absolutely not there. And this technology in past 10 years, for which very quickly the Nobel Prize was given. So not only we now know the blueprint of the life, we can also modify it at our will in very precise manner. And that's why the genomics is taking the center stage in various things. And just to impress you about the scale of the problem though, if we are entering in the genome era, we must understand what is in it. So each one of us carry enough DNA that you can make a that you can make a trip from Earth to Sun 60 times. 60 times round trip, that much DNA all of us are carrying. So that is the size of DNA you all have in your 50 trillion cells. Right? So that's a huge amount. And 3 billion is the size of the human genome in each cell. And as you can calculate it, we have 50 trillion cells, how much it will be. And this 306 meter of DNA in each cell is packed between the 10 micrometer diameter of a nucleus. So it's hugely packed. This big thing is packed in even invisible amount of thing. So the DNA is very twisted, wrapped, folded. By the time you reach here, you can't see anything. You just see chromosomes as they are. So the big question comes in how this packed material is transcribed very precisely, that liver cell will produce only liver-specific mRNA, brain cell will be transcribe only brain-specific mRNA. They will not suddenly transcribe and express protease, otherwise that will digest the whole brain, although gene is there in each cell. So it's amazing regulation that happens, and it's one of the challenging areas that people are studying in the area of developmental biology. So how this information is accumulated, stored and maintained and is expressed into in the DNA-based information system of life is a big question which will keep working. Just to give you little bit more of a impression of how we look at the DNA is that all life information is encoded in genome. In the little egg that you see from a chick comes out is because the chick DNA is there in its egg, and it directs how to make from single cell to a bird. You can see there are little eggs laid by the butterfly. They become larvae, they become bigger in size, they become pupae, the larvae, and then one day they will become a butterfly. Same genomic information that has this lifestyle and this lifestyle. Here the feeding, the escaping from the enemies, technology is completely different. They look like different animals, right? But the same identical DNA sequence. So these are very interesting, challenging questions of biology. From the one seed, you will see one huge plant, a tree will become, and that will live for hundreds of years. So these are the challenges. If all this information is there in genome, can one day, looking at the DNA sequence, I told you, we can very easily now read the DNA sequence. The technology has enabled us. Any DNA you give, any animal, plant, I'll give you the whole genome in few days. Can we read that DNA sequence and tell the owner of this genome what is the shape, size, behavior, response to biotic and abiotic factors, disease susceptibility, longevity? Right now we cannot, unless so that means we haven't understood how to read the DNA sequence, how it gives the information as it translates itself in the nature. One way of doing this is to compare the genomes. So if let's say, and then look at the differences and associate that to the characters. Suppose these people have, let's say, curly hair, and these set of people have non-curly hair, straight hair. Now I compare the genome of everybody and see that what is the one single thing which is common in them and is missing there. That is very likely the character for that is leading to the curly hair formation. So this very simple way, if you have many different organisms, you compare the genome, we can start reading and associating function. Then of course, if I go and change that particular locus, they should also start getting curly hair. Then I establish this. So mechanism is there to study these things, but as you see, that's challenging. So here I give you some example. If you take a simple organism like yeast, and if you go to organism like human, the genome size increases because you need more information to make a human as opposed to make an yeast. But if you see the number of genes, that is not increasing so much. Yeast has 6,000. By the time you reach C. elegans, which is worm, you already have 20,000 genes. And then onwards, the gene number is almost fixed. So that means, similarly, the percentage of genome that is coding for proteins is very small. In the human, it's less than 2%. Right? So the gene numbers is not changing so much. What is changing is the non-coding part of the genome, which is reflected here also. Like humans, one gene requires 120,000 base pair of DNA around the coding and around it, because it has more number of two cells. Our body has 210 types of cells, while Drosophila have something like 50, 60 of them. So that is how we measure complexity. More complex organism, more different cell types coming out from the same genome. Genome is identical in them. So in nature then, the number of genes have remained static, but more regulatory sequences have been recruited to give the same genome many different functions, which we'll call functional forms, we call the epigenomes, or the functional versions of the genome. Okay? So that means we have to look well beyond genes to understand the biology of the genome. So, so that is about the genome, the challenges and how it does and how interesting and exciting it is. But now there are technologies that are coming which are setting up the future, whether it's agriculture, whether it's nutrition, whether it's healthcare, everywhere genome is going to be very, very crucial. And these technologies are that we can sequence genome, affordable. I'm sure soon human genome is going to be sequenced in less than 10,000 rupees. So all of you can go and give your one drop of blood and get a whole complete genome profile and start using it. We can do at a scale. You may have heard recently DBT announced 10,000 genomes were sequenced. Before that, CSIR has sequenced 1,000 genomes, and now millions of genomes are being sequenced. So we can sequence actually the whole population now and sequence it fast. Earlier, genome sequence would take months and months. Now it takes days. And there are now computational tools that can compare the genome and analyze genome functional analysis, data analysis. Huge computational resources are available. And as I was saying, the genome editing is the most powerful. You know all this information, you can also change. So you can efficiently change. It is affordable to change. All of us are using in the lab. This technology is very accurate and it can be done with very little cost. So all these technologies are impacting our healthcare, sustainability, nutritional security, environmental remediation, industrial applications, and of various kinds, biosecurity, all kinds of things. Now, if you know the genome, you can make a virus which can create problem for people, right? Or problem for a particular crop in a particular country, if it is unique. So these are huge biosecurity problems that are coming. So that means you have to, for your own survival and for your growth, you need to use these technologies in a very efficient manner. With this introduction, I will show you one slide. All of you are aware of CSR system and CCMB, where I spent many decades. So now I'm working for the institute, and the work I'm going to explain to you, we are doing in the beginning, at least environmental surveillance in CCMB, which we have extended here. So TIGS, you might not be knowing this. Tata Institute for Genetics and Society, the trust where Mr. Ratan Tata is the settler. And this institute is set up, it's a private non-profit institute funded by the Tata Trust. Of course, we get funding from many other organizations, including DBT, DST, ICMR is our major funder. So the vision of this institute is to use cutting-edge science to solve societal problems. We are a program-driven institution, not PI-driven. So we choose a problem and solve it. And each project has to have direct societal relevance. We don't do for knowledge generation, for publication, for training. We solve problems. Everything else is collateral. So that's a little bit different kind of thing compared to what many other academic institutions do. And we have 3 programs: infectious disease, where we have vector control. We have actually world-class mosquito laboratory. When you guys come there, you can have a look. We are located in GKVK campus, North Bangalore, in the same campus where NCBS, INSTEM, and CCamp is there. So we are part of that campus. And we have crop improvement, where you are using genome editing technology, mutational breeding, and environmental friendly pest control. And rare genetic disorder, which is a major problem in the country, which not attended by many industries for a variety of reasons. So we are addressing that. So these are the 3 major programs, and we do projects in that. Looks like it's okay. Yeah. So now I'm going to give you just few examples. These are very broad examples. I'm going just to really excite you and catalyze your thinking in those directions. And I'll be very happy to discuss. You can do Google, you can talk to me. We can, after the talk, there will be some time we can discuss. You can write anytime if you are interested in any of these things. This is just to really expose to you what is coming in future, and coming not only in future, you can also do. These are the genome technology. The biggest advantage is you can also do. It doesn't require huge setup. You don't have to set up DNA sequence. You can get things, a sequence from somebody, and then do what is required to be done with that. So there is technologies in the biotech revolution. One of them, okay. So think of a designer crop, right? This idea is there for some time. So think of a crop which is a pest resilient. So less or no pesticide required. Yeah, so I'm trying to. If you do it. Oh, I was going, okay. Oh, okay, thank you. So disease resilience, so it's many times we even don't recognize those disease. Climate resilient crops, because you can see the temperature going up far more than what was anticipated, unusual rain patterns and pollutants of horrible kind. And then sustainability in our agriculture, we will not have water to grow rice after 10 years with this much fresh water. 1 kg rice, you require 3,000 liters of fresh water to grow. That's not tenable. So we need new methods. Then also you need high protein, because we have lots of food but less nutrition. Most large number of people are malnutrition. They don't get enough protein, enough vitamins and other nutrients. So that has to be brought in. So higher protein, higher nutrients, lower anti-nutrients, and lower glycemic index. These are, if you have to design a crop, these are the things that you will look for. So you can address now the stress on agricultural land by keeping all these things in account, sustainable nutrition security, and making natural organic farming viable. So if we have these things in the on our cards, how we can make a crop which is yielding high, although we have enough crop, but if we have 20% more yield, then we can use 20% of that land for something else, or let others animal use that, because they also are members of this planet. So, and imagine no use of pesticide, no or less chemical fertilizers, and so on. So I'll give you some couple of examples. This is just to show you that a small institution like it is. We are just a 6-year-old institution, discount 2-3 years of pandemic in that. We are total 120 people, 20 scientists, and if we can do, then everybody can do it. It's an easy thing to do. So in crop improvement program, we are aiming to develop crop varieties with desirable traits, which is again disease resistant, climate resilience, high nutrients, and so on. And we are using technology like genome editing. Genome editing is not in a GM, so it doesn't go under GM thing. It's considered like potential mutant, so it can it is more availability. I forgot to tell that everything that we do has to have 3 components. A, if you give me a problem, so I will first see that. We'll see if there is a science available to solve that problem. We will bring it from wherever it is required. 2, the solution that you are coming up has to be economically viable, otherwise it's not a solution. And the third, it should be socially acceptable. Otherwise, if people are not accepting, there's no point. We don't want to spend time in Supreme Court to get something done. So we do things which is for which the pipeline is there in place. So we use it, that's why we use genome editing technology, although it takes time to improve the traits. We do mutational breeding. This we are doing in collaboration with CCMB. You create mutants and look for the varieties. I'll give you some examples. And we do pest management. We work with the Karnataka government and GKVK to find solutions to some of those things. So the genome editing, our targets are pathogens, whether it's a bacterial leaf blight or a fungal blast or insect pest, brown plant hopper. So all these problems we have addressed, and we have now varieties, genome edited varieties, which are taking care of these things. And then for the weeds, the herbicide tolerance, and that also allows us to grow in less water, various kind of these plants. So there is one example. Here is the resistant plant which is not infected, and susceptible plants can get infected, and these are the assays that you can do in the lab. So whatever we do, we do as a technology platform. So in a sense, once we know how to do genome editing, then no matter which locus is required, you just change those primers and guide, and rest of the thing is established and fixed. That doesn't change. So that is how we have now set up a timeline for everything, how much time each step requires, and we can measure. So like our bacterial blight lines have reached up to RCGM approval. This will go to field trial in the next season. Then other multiplex gene edited lines are somewhere here. They will get multiplexed one by one. And we are also just begun millet and pulse genome editing, setting up the genome editing platform for these 2 important crops, and then introduce like in millet, we want to increase the grain size because it's very difficult to harvest, reduce the anti-nutrients, and increase the yield, because we talk so much about millet, but millet is not very profitable for the farmer. That's why they don't grow. But we want to convert and increase the canvas of our nutritional plate. In the mutational breeding, you take the rice, go to a gamma radiation facility, radiate it, and then start screening, establish lines. So we established thousands of lines. So we have done some in the collaboration with CCMB, some we have done here on our own. And we look for the higher iron, zinc, calcium, magnesium. So we have set up this facility to screen these things in the lab. We have, we call it NuGen facility. So for the zinc, iron, and all these metals, proteins, glycemic index, we can measure in the lab at a high throughput. And we also look for anti-nutrient and better bioavailability of nutrients. So like here is one example. So we take some Masuri line and do the mutagenesis and screen large numbers of, you have to screen thousands of lines, and some of these which are showing higher proteins. So rice, 7-8% is what you have in the rice protein, but you can see you have pretty high levels of, if you can establish this as a line, then it solves the problem. But then there are many other things where labs like CFTRI can be useful. When it becomes more protein, then people don't like that rice. It becomes hard, it doesn't cook properly, and taste is different and all. So we should think of now how to make that thing acceptable. But this is work in progress. We will keep growing these lines and seeing that they are stable. So here this is again, which is of interest here. Processing and cooking can do lots of things about the nutrients. So here is the zinc. If you start out with this, you do brown rice, polished and washing. You retain much of the zinc, very little. But as far as iron is concerned, by the time you polish and wash, all the iron is gone. So even if you have high iron content, it's not inside the seed, so it doesn't help us much. And again, here you can see that iron which is there in unpolished rice, after polish it disappears. So we have to find ways of processing the food that it doesn't use its nutrient value. Here is an example of looking for the zinc. As you see that different mutants that we have collected, we have grown them in different seasons of different years, and they are retaining their high zinc potential. These are the things, different generations we are taking wild type and mutant, and you see mutant is always having more zinc. We go in Hyderabad, in Bangalore, and check if there is a climate difference and those things, so this is stable. So looks like we have higher zinc producing lines, which is quite stable. I'm just showing all these few of these things to show that if you think properly, now technology is very easily available. You can do any genome editing in rice, or if anybody interested, we can do. It's a very established thing, and you can go and take try it out in the. Several generations. Then there are, it's working now? Yeah, thank you. So then there are more very high-tech, fancy, not practical at the moment, ideas like lab meat. So like you heard in the beginning, we did a project with DVT and National Meat Research Institute in Hyderabad. We could grow the meat in the lab. You can scale it up, but only thing is very expensive. Because to grow those cells, you require the medium, which is very expensive. Growth factors, serum. If you have to use serum, then whole purpose is lost, right? Then you have to anyway kill the animal to bring it. So there are efforts happening to make those constituents of the cell culture by chemically synthesizing or maybe adopting from the plants, and then growing the cells. So, strategy of our India should be to keep the technology, and when on and keep researching on this, and when this becomes viable, then implement it. But for this population at the moment, from considering environmental factors, for bulk of the population, plant proteins are the solutions. The other solutions will be insects, but then there has to be a lot of cultural change has to come, that people accept insect protein to eat. Or you find a way of use plant make insects, then from there meat, or lots of husk and other secondary products from the crops are wasted. You can grow mushroom in that. That's a really viable option, or the algae and processing. You can take low cost proteins and process them and make high quality edible things. There are transdisciplinary university in Bangalore. They are doing lots of good work in this process. They make dal from various things, and that's actually even they have tested, and it looks like very interesting. So there is a lot of scope because you have to feed the world's biggest population, which is only increasing, and there is no solution that somebody else can produce food and which we can sustain. We have to make food. We have enough quantity and a quality. So that was about the food. I am going to speak of two other things where the genome technologies are going to have high impact. One is therapeutics, another will be environmental surveillance, which all of us are part of. So we'll go quickly. As I said, you don't be stressed. You don't have to worry too much about any. I'm not going to talk any deep science. It is just there may be some terms which may be new to you, but is very, very simple problems which genomics is really making impact, and some of them I will show you which even we are able to do. So in the healthcare, there is a huge economic potential of genomics. So like global gene therapy market is $6 billion, rare disease treatment market is $200 billion, global genome technology of just creating the data and editing another $50 billion. So these are huge things. One example, if you take rare genetic disease in India, we call it a rare genetic disease, but we have any given time 90 million people. For most of these diseases, there is no treatment. If it is there, it is not affordable to anyone of here. And this is lifelong disease, disabilities. So you may be knowing many families because certain population in south, even in north, and minority communities, certain who live in small numbers, rare genetic diseases are becoming not rare. And so family goes through lots of disasters. So we have given a thought, what are the affordable options for this? Can we have mRNA therapeutics, in vivo gene therapy, and other novel drugs? Because it's not only that you have 90 million people, 1 million patients are born in India every year. So that's the number we are talking about. Can we prevent that birth by genetic screening and genetic counseling? So here just there are 7,000 plus genetic disorders. All over the world, if you count, it's more than 350 million. So if you see that, if you put all these people in one, this will be the third biggest country in the world after India and China, right? So that means it's a very big number, but they are totally ignored. Hardly anything is done. And some of you, if you know a person who has muscular dystrophy, various kinds of neuromuscular disorders or blood disorders, it's really, really bad. So what we have decided to do is, and as I'm saying, these are very simple things. You don't require big infrastructure. So we set up a technology platforms for mRNA-based technology platform, therapeutic platform, and cell-based therapeutic platform. So platform means it's a disease agnostic. Once you set, it's like a bag making company. You can change the design, rest of the machine, material, everything is same. You can take different style of bags. Only the beginning design, and maybe later customer will be different. So that is one, and then it becomes economically viable. So you can give solution for not one disease but 20 disease. So then if the number of one disease is less, you are economically viable. And you can, once you develop one, then you actually have developed everything, right? So that's why we do most of things in a platform technology way. So we have used actually, even in CSIR, we were trying to make a mRNA vaccine, which Moderna was making, and we were in discussion with them. But Moderna was not interested in doing this vaccine in India, so we started making on our own. In fact, we made mRNA vaccine, which was tested, we were working against hamsters. It was tested in ISC, and then a company came. It was handed over to a company, but by then COVID went. So, but that gave us confidence that this is very easy technology you can develop. So now we have we are doing some vaccine, but I'll give you some examples that you make a mRNA, is very easy to make, which will make therapeutic protein. And then you have ionizable lipids, mix a certain composition of them, put them together, and you make the lipid nanoparticles. And these are then injected in the body, and that will, because this mRNA codes for protein, it will make that kind of protein, and then you can go to humans. And these are the lipid nanoparticles that we are making in the lab. So, and if you inject anything in the body, generally it ends up in the liver unless it's targeted somewhere. And it so happens that many time diseases that are there, liver is the target for them. So you inject, and this will be endogenized by LDL receptors, and it will go through endosomal trafficking. It will then translated protein will be made. Protein is tagged, so it will be excreted, goes in the blood, and goes on to its target and works. So we use liver as a manufacturing factory, and to distribute the rest of the body wherever the protein is required. Right? So this is relatively possible to do. Why we have decided to do this mRNA therapeutics is that, so these are, you don't have to go into detail, but cost of one dose of Moderna vaccine is $0.2 per dose. Okay? So that means if we make in India, it will be just few rupees. Rare genetic disorders, lysosomal storage disorder for which the treatment is available, per year treatment for a child 10 kg or more is 40 to 50 lakh, and bigger the more the weight, up to 1 crore per year for lifelong. How many of our people can afford 1 crore per year for treatment of a child, and the rest of the household remains to be done? So that means those are not affordable. But this will be a few hundred rupees, in maybe 10,000 for a whole year, the treatment is done. That's why we decided to do, which will be a disruptive technology and will solve the problem for the people. And the mRNA therapeutic is very powerful because protein is now, normally enzyme replacement therapy is that you make the enzyme in the lab. In fact, it is imported, very expensive, and inject in the blood, and then how well it is folded, PTM is good or not, and protein will 80% is degraded within few minutes. But when the mRNA is injected in the body, then it will make, the body will now make protein as if it is its own thing, and then it will be properly folded and modified and so on. This is a very, very powerful technique. And we have established this platform. So one stage, first stage is the mRNA design. Once you know the DNA sequence, the other things are almost in the place. In a month or two, you do that. Then validation means you make, once you have designed, in 1 week you have the mRNA put in the LNP and infect in the cells, and put in the cell, and all the iPSCs of human cells. In couple of months you know that is expressing or not, and then you inject in the mice and then see that mice is happy and protein is being made. So in 6 to 12 months actually your preclinical stage is done. Now all you need is go to drug controller or ICMR, and this goes through GMP facility, goes to people. So you provide solution in months, not now in years. And just to give you some examples of what we are doing. So we are doing these 4 different diseases, which are 3 are lysosomal storage disorders, and 1 is monoclonal antibody. I will give you an example that how these are the tremendous opportunities. So you take the therapeutic thing, inject in the patient, liver will make the protein, and it will go to different organs where it is required. You can also do targeting, which we are doing for the GNE myopathy, because very specific muscle is what is affected. So you provide in the thing, and this will go and express in the muscle, and then you provide the protein, and patient starts to walk. And this is just to show you that when we inject in the tail, the protein is expressed for several days in the mouse. This is the liver of the mouse where the chemiluminescence is detected. So that means, and this mouse is happy. So here is just to, I'm not going to show you any data. This example, there is a disease called Pompe disease, which where the gene that is affected is GAA. So we make this GAA mRNA, make lipid nanoparticle, put in the mice, and this is expressing nicely, and for several days in mice. The same way you see here, Fabry disease, because it's a platform, you just have to change the DNA sequence when you are making the construct. Everything else is automatic, is done immediately. So these are the diseases for treatment cost 1 crore per year, and this solution that you are seeing here will be few thousand per year. So that is the power of technology that is going to change the way we are doing therapy right now. This is another big example. If some of you are interested to become entrepreneurs, monoclonal antibody is one of the big hit in the therapeutics. But 80% of this is used in the rich world, only 20% is used in countries including India. Today the sale is 125 billion, but most of the patients are here where they cannot afford, so it is that kind of sale. So what we have done is that one of the, so what you can do, you can, instead of this mRNA now can have a heavy and light chain of the antibody, because antibody is known, the monoclonal antibody. Once you do package it here, put in the mice, and mice will, if it is working well in mice, then you go to humans. And so here is the example. So we did the same thing. This is the trastuzumab, which is a antibody which is used against cancer and is after patent. So we took it. Now we did the same thing, put in the mice, and is expressing nicely. Mice is happy, and you can see for several hours lots of protein control. There is no protein. Then BC and BD, they are two different combination of light and heavy chains, and the mouse is expressing this. So that means, and each injection of this, if you go to hospital now, and if unfortunately anybody you know needs this, it will be between 1.5 to 2 lakh per injection. This will be few hundred. So there is no comparison, and then only India can afford. How many of us can afford 2 lakh one injection of a drug? 90% of our population has less than 25,000 per month salary. This is not possible to for them to afford. We live in very different world where very small number of people have lots of money, so life looks simple. But we are not. The India is very different from that, and these are the solutions for our people. Here is the vaccine part. So you can again design a vaccine, multivalent, and test it. In 2 months you can have a vaccine. So since vaccine market is quite crowded, there are lots of companies. So we are working for the animal vaccines, leptospirosis with Nivedi, which is a Bangalore lab, ICR lab. They have a specialist in this, and in just a few months' time, we could make this vaccine and give, and they are now testing it. So I'm saying you can have a very small infrastructure. The technology enables you to do things which were actually unimaginable. Here is another example. You can do gene editing using the same technology. Right now, I am putting in lipid droplet mRNA, which goes into the cells and makes protein. Now, instead of that, so we have focused a few diseases here. You take the mRNA for Cas9, which is a enzyme that will cleave the DNA, and then you put a guide RNA there, and the template, depending upon what editing you want to do, or the whole protein mRNA you can put. Now this whole package will go open up in a cell, it will go into the nucleus, guide will take the Cas9 to a particular site where it will cut, and the repair system will use the template that you have provided and fix the problem. So with fixed problem, that means single injection, now the mutation is corrected and the patient is fine. And depending upon which tissue you target, you can do correction there. Or you can whole protein which is missing, let's say here is this exon gets mutated heavily in this disease. So you can actually remove the whole exon and provide the entire patch here and fix the problem. So this is for the DMD patient. So these are the things we are doing in the lab. So this will not be like mRNA therapy or enzyme replacement therapy. Those are once in a week. This will probably be single shot or 2 shot in whole life, or maybe annual dose. It will be cost effective. Again, cost will be few thousand. It is not in lakhs and crores. Gene therapy today cost, one-time gene therapy costs $3.5 million, which means 20 crore. So 1 crore per year or 20 crore in one time. None of these are possible. This gene therapy will be few thousand, because it's just, you don't have to go through the gene therapy if you know how it is done. It's a horrible process. Here is just an injection given, that's all, like any other thing. So you can actually these technologies adapt to our need. And so here is in the lab that we show that if you take cell culture, we can do 70% editing. So you put this whole thing and then see how many molecules of the DNA got edited. And when you put in the mouse, the same thing, as I said, this is platform is very fast. We can see with 50% repair, editing is done in the liver cells, and that we are targeting a disease which will reduce this is for the familial hypochloremia. If you correct it, it will be a very simple treatment for the patient. So these are things, and our mRNA therapeutic team is, you will laugh, is one scientist and some 6 other people. These are 6, 7 people in a lab of this size in NCBS 1 floor. All this is happening. So these are so simple to do if you are interested in doing. Here I will not talk much. This is a human-induced pluripotent cell technology platform where we have established that you give a blood, 3 months, we will give you a iPSC. You can now grow an iPSC in a disease. We can use CRISPR technology and do editing and make a disease model. So you can have an organoid or a disease model in the plate, and you can do your drug screening or understand the disease more and do a straight away trial in organoid and animals, and then go to. Same way you take the patient samples and can convert the organoid of the patient and then do the trial of the drug exactly for the same individual. Right? So these are the personalized medicine things that you can do, and these technologies are established. And this is where in the therapeutics, I gave you a few examples. There are a few more things where the treatment is going to be many tablets are going to be replaced by little injections which will fix the problem, no side effects, because body will take care of itself and not the chemical which will go work on 100 organs, 100 targets. One is good one, 99 is a bad one, and you adjust the dosage and so on. The last, maybe I'll take 7-8 minutes. So, is about environmental surveillance. This is something even I was amazed when in COVID time we began this business, and this is something really amazing. So, there is something called environmental DNA. All of us shed, each one of you every day shed 330 billion cells. You just leave them through your intestine or through the skin, and these cells, you are distributing your DNA in the environment all the time. So if I have vacuum, strong vacuum, after you all go and I do the vacuum of this room, I can genotype all of you because you have put enough cells here. Right? So what we have done is we have started doing wastewater surveillance. All of us do whatever, in the end all the material ends up in the sewage system. So whatever we excrete, we sneeze, do all the surface washing, everything ends there. So in the environment, so if humans do, humans are not anything unique. All the animals do this. So that means environmental DNA represents what is there in the environment. Without looking at anything, you can actually take this DNA, and all the microbes that you are seeing will come here. And that is why it brings in the context of One Health, where human, animal, and environment, which is including the plant, you can measure their dynamics with very little effort and cost. So you can do, if you can take environmental, some STP of your institution, if you take, I will be able to tell that how many kinds of animals are in this campus, how many of you are carrying flu today, how many of you are carrying SARS-CoV-2, because all this will be reported in the system there. So you can detect microbial diversity, you can detect pollutants that are there in the environment, the ecosystem healthy, this way you can put. And these technologies are now used for, let's say, metagenomics of marine environment system, soil and agriculture applications, urban and industrial assessment, and so on. So the metagenomics is when you are taking and sequencing like crazy and then doing bioinformatics and extracting. But if you are monitoring particular disease, then you can do RT-PCR and detect how many, like we have been doing for past now 4 years in 28 STPs of Bangalore, and we know which zone, how many people are infected with SARS-CoV-2. Even today we are doing still, there is no symptom, everybody is vaccinated and happy, but we know the peaks that are coming and going and how many people are infected, and now we are also extending to other diseases. So this environmental surveillance gives you preparedness, that how many people are infected, what kind of disease is increasing, what kind of disease is decreasing, is there a correlation with temperature, with the population, with the festival, with the climate, with the rain, anything. And so that if the system is ready, and we work very closely with BBMP, happy to with Mysore if it is possible. So the civic system can have a strategy to take care of this problem before they become a disaster. So here is an example of SARS-CoV-2 monitoring for several months. This is the first wave, then second wave, then third wave, which was Omicron wave, which the red is the cases that are tested. So after this, there is no testing done. So we see peaks in environment. These are different variants, and these are STPs that we collect. So that this is just to show you that we have very precise idea of how many people are infected. So this is from the STPs. Hyderabad, somehow government was not cooperating. They were not giving access to STP. So we were taking open drainage. That is, nobody can stop you from collecting water from a nala, right? So but that was lot of work to what time, which place to collect. And this is now very interesting. We are still continuing to do that, and you can see the same kind of peaks coming here. There are some 20 open drainage system we have taken. Most of the country, anyway, there is no STP. So you have to standardize technique which works for smaller cities, big cities. So this became actually a very strong point for us, and we have now proposed to the government, and is very high level now. NCDC is considering. We have set up a national guideline committee to implement it all across the country, so that we know what is the situation. So whether it's like Bangalore example of STP, Hyderabad example of open drainage. We have also done air surveillance in CCMB. Now we are doing very serious level in ticks. And idea will be to link all these things to the vector surveillance and to the clinical surveillance, and provide a full view of pathogen situation of that particular city or the town. So when we were doing this, when COVID became irrelevant, but we had generated the capacity, we started using, this was a very big grant funded by Rockefeller Foundation, some $7.5 million. So they said that you can do with the AMR. So we, Bangalore, Hyderabad, Pune, and Delhi, we started AMR surveillance. AMR is antimicrobial resistance, is actually a huge problem right now. And in few years, all our antibiotics are going to run out of any efficacy, and there will be already millions of people are dying, and the estimates are that by 2040, 50, there will be 10 million deaths per year because of this. And the AMR comes because use of antibiotics in the poultry, some ways in the plant, in hospital, people use on their own, and everything comes, enters, antibiotics reach here. Bacteria will thrive, because now you are putting selection pressure. Whatever is resistant to antibiotic will grow, and then they will transfer the lateral transfer of genes, and then they will, even if it is used for livestock or pet, it will end up in the human. So it's a huge, huge problem, and WHO is terribly concerned, and even the ICMR is highly concerned that how to solve that problem. And so this is a global issue, because if resistant bacteria will go everywhere. So there we employ the environmental surveillance. We collect environmental samples, process them in the lab, and then do the data analysis and come up with the whole profile. And this is just to, I will not show you a lot of data. This is just to show that these are 16 different open drainage systems of Hyderabad, and you can see there are a lot of similarity there. One kind of macrolides class of antibiotics are the maximum resistance, one is exception. These are very interesting. This was a site which is just downstream of a slaughterhouse. So the lot of thing is not animals, humans, there are animal AMR is coming. So this actually gives you lots of things. So this data we have plotted for which mechanism is operating, which antibiotic it is, and whether it's changing with time course, and so on. So lots of information is coming, and ICMR is very excited about it. This is a very scary picture. So this is like E. coli. You can see E. coli. These are different antibiotics that is they are resistant to all this. and these are various kinds of other pylori, pneumoniasis, all kinds of bacteria, and how many antibiotics. There is no antibiotic left, except one or two. So, because of increasing population, expanding geographical areas, climate change, close contact of the people, deforestation, so animals come to human thing, disease outbreaks, all that, and misuse of antibiotics, all this is leading to increase in antibiotic resistance in a very, very massive way. And the only way to do is to do environmental surveillance, at least to understand what is happening. But there are challenges where you want to collect the sample, STP, open drainage, water body, river, city, village are different things. How often you should do, every week, every month, what organisms to look for, and data sharing and using in a strategic way or safe way is another issue. This is one example of a US city where there is a sewage system here, and there is a river, and river microbes near wastewater treatment plants express high level of antibiotic resistant genes. So, this is that dangerous that I mean, you will imagine that country like US, it will be done very well and all that, but nowhere anybody is safe. So, we are talking about one health, one planet, and it will sustain only if there is a collective effort from not only different cities and states, but different countries. So, we have now joined hand with ICMR. ICMR fully funded this, so they were very excited and impressed to what we showed in few cities. So, now we just last month we started this project for AMR surveillance through VRDLs. I am sure there is VRDL in Mysore also, virus research and development diagnostic laboratory centers, where we do surveillance. TICS will do metagenomics and provide the kit, and then VRDLs will do AMR surveillance, and we will compare the data and show what kind of VRDL is existing, and how this antibiotic resistance is coming, whether it is coming from poultry, is coming from livestock or from medical use and so on. This is the last slide to show you in which way I am very excited about this, because we are doing things now a little bit differently. We do science for the society, right? And this is very challenging science, scientifically very, very challenging. It's not that it's anything less challenging than basic biology, which I have done for the rest of my life. So, you have to engage the community. You have to take the technology, make technology acceptable, accessible, and taking science to people and do policy advocacy. When you have, like we have now solution for rare disease, but our system doesn't know how to do trial for that. So, we are working, making policy, working with the regulators, with ICMR, with the CIDSCO and the other centers to see how we can take this, make this solution reach our people. So, we do a lot of community engagement and policy stewardship. We have social scientists as our faculty. Our focus is to translating science for societal needs. As I said, economic viability of the solution is important. If it is not affordable, we simply will not do it. And any solution we provide, if the same solution, because we are non-profit organization, our cost is going to be 80% less, right? Because 80% is R&D cost of most of them. You indigenize on that, the cost is a further 10 times less. So, without any major innovation, you will sell things at a 10-fold cheaper. But we want 100-fold or 1,000-fold cheaper, then only it will be available for our people, and that is where innovation and research is required. So, we are doing lots of, like for rare genetic disorders, we have chosen population, where we do population screening and do genetic literacy, because this can be actually avoided, and involvement of people is very important. And I must acknowledge CSIR and CCMB, where all these things had begun, where we are still collaborating. ICMR, which is our major funder, and Tata Trustees of course funds us very generously. DST for giving me J.C. Bose Fellowship, so I can keep my students active. DBT has funded our RISE project. Rockefeller Foundation has funded the surveillance program, and ACSIR is TIGS is also affiliated with ACSIR, so we thank them, and thank you, and I am very happy to answer any questions.
Thank you, sir. And the dice. It's open for questions. Any students can ask questions.
Now, is it possible for us to think about other varieties to bring low glycemic index rice? That is one. Second one is, I mean, all over the world, people are struggling how to increase the iron content in rice, but they are not reached. More than 4 milligrams they are unable to increase. Another one is, yield of pulses in our country is very less, around 18 million tons, whereas Canada produces a little higher, I believe.
Yes.
Now, by the genomic technique, is it possible to increase, or whatever I said, something is possible to attend in that direction so that to increase the pulse production and iron content in the rice and some other glycemic index, please?
Very relevant questions. Thank you for asking. And all the three are actually doable. So, the low glycemic index of basmati can be brought into by simple breeding technique. So, we have to keep monitoring, or if we can identify genomic locus, then we can go and straight away edit the locus and bring in. We have like, I don't know about basmati glycemic index, but the improved Sama Masuri that CCMB and IIRR and agriculture.
Now, if I am correct, it is 32 something, or 32 or less than 40 glycemic index of
that
basmati
will
rice.
be really, really surprising, because
that's what-
something
that is what the
44,
literature
5
says
is
like.
considered very low. That will be something, I'll figure out if this is the case. We will focus on this to find out what is the thing that is responsible for that. It should be possible. See, all the traits have some genetic locus. If we can identify them, then by genetic crossing or by mutating, either way it can be done. Iron is a challenge. I showed you that we have made varieties which have more iron, but this iron is only in the coating, and if you polish, then it goes away. So that is the problem. Brown rice people don't like to eat. Even I tried eating, but after some times you don't feel like eating.
But even if you take low polished rice, around 3 to 4% polished rice, that iron content remains with, that is a great boon to the society.
So it should be possible to increase that. So as I said, when we do mutagenized so we did experiment with 1,500 lines, but I think we have to do tenfold more. So we have to say maybe 100,000 lines, and screen you might find something. These pathways of these things are known, so as the experts have suggested us to do certain genome editing, so those are also possible to do. Your third question was about
yield of
yield of pulses. Yeah, that is very important. That is, there is a lot of, so yield has many factors. First of all, the soil, nature, the kind of nutrients you use, kind of practice you do, and the variety is not only pulses. Many things like soya bean, many other things, China or other European countries and Canada, they have 2 times, 3 times more yield than what we are having. So we are doing very inefficient way, and that needs to be fixed. And this is very much possible. That's how we decided working on pulses, because that is very important. So one is, of course, to do a genetic approach and improve the yield. But I think even some agriculture practices are can be brought in, and yield can be addressed to quite a significant level. But I can tell you now, in the current stage, because rice, there is a lot of study happening here. I have seen in IIRR genome edited rice line 20% more yield than the top yielding variety. So that's amazing. That means you will use 20% less land for the same yield. But imagine that farmer has to deal with less area, less water, everything becomes 20% less. So that will have very compounding effect. So yield is an important thing to look for, and it's certainly possible.
So another just clarification, sir. This is among the rice varieties, we have colored rices and brown rice. Generally, colored rice, what are available in our country, wherever you go, all rices are bold in nature. They are not fine in nature. So table rice point of view, that rice becomes a negative. But at the same time, phytochemicals wise, otherwise, it is having high nutrients, also very high in the case of colored rice. Now, is it possible in this direction to think about colored rice in finer varieties? That's a very
anything,
challenging
the,
point it is.
yeah,
If it is
so the
comes out, it's a very good.
so biological system is such, anything is possible. After all, that is also because of the genetic structure of the rice is there. But while you can do that, the one something which is maybe very relevant to CFTRI is that actually you can develop processing of those rice and make the processed version. People are making remaking the grain of rice, right? They do graining of pulses, rice, and they will look like rice of different shape and size, whatever you want to make. So that is artificial way of doing, but I think while making shape, and so we are all evolved to eat something, and that is our inherent nature. Animals don't analyze how much nutrient is there, and we have evolved because that was nutritious also. Because of the now many other environmental factors, that habit is not giving enough results. We are not eating healthy food, and we cannot change habit that easily. But I think processing of the food is a very important aspect which can be used. As I was saying, this university is making pulses from collecting proteins from various things and making pulses which will be half the cost of pulse, is 40 rupees a kilo they are saying, and protein is 30% in that. And it can be, it will get cooked in 5 minutes. Yesterday only I was discussing this. It's so amazing. And actually, if it is taste is also good, it will. Anyway, half the time we are eating food which we don't know what we are eating. It comes in a packet, and we are looking at TV and eating without knowing what we are eating. So we can use all this new human nature and process food in a way that is nutritious and safe and sustainable. Yes.
Thank you so much, sir. You've covered a very broad spectrum of topics
Yes.
in your
Yeah.
talk, actually. So I have 3 questions. First thing is about the evolution of non-coding RNAs.
Yeah.
If you compare the yeast and us, we also share a lot of proteins like GAPDH or glycolysis, everything. And if you look at non-coding portion, you have different kinds of non-coding RNAs like pseudogenes, link RNAs, antisense transcript. Is there any preference for them to amplify a particular species in this, like they selectively amplify, acquired more pseudogenes, or anything is there through evolution?
There is no particular, see, when a new mechanism evolves, a new branch of animals, so what you said, some things are very basic, like all cell cycle genes you can take from yeast and use in humans, and vice versa, it is the same gene. Functionally the same, sequence may be a little bit different. But as you go more and more, you acquire more and more regulatory features.
That's
the
right.
same gene now will be expressed in very sophisticated manner in response to certain environment and other, and that's what has been added. So what I talked about is that the non-coding DNA has been added, which does regulation. Some of this non-coding DNA is transcribed, and RNA is as the user regulatory code, not coding for the protein. So these are something which like every is very interesting. Like in this process, there is something called RNAi pathway, like RNA. In fact, we are using this technology for insect control. Yeah, sometime we can discuss. Like, but yeast, cerevisiae yeast, the budding yeast, does not have the entire pathway. But while the fission yeast, Pombe, has that pathway. So there are life has many possibilities. So these are things we are different because we have chosen different molecules which have become important for a particular species, and within a species there is variation. So although I am giving you very vague answer, but the thing is it's very, very complex, and there is no rationale that this thing is selected in humans. This thing. Like one of the DNA sequence we identified very early was we call ultra conserved sequences. So ultra conserved sequences are DNA sequences which are conserved 100%. 100% means 100%, several hundred nucleotides. Not even protein coding requires. Every third nucleotide you can change for the identical protein. And these we found they were affiliated next to the homeotic gene cluster. That's how we found. But it turns out that there are several thousand of this in human genome, which is identical from fish to human. So that means 400 million years, nothing is changing there. And it is still a mystery, why so much of conservation? So one of the aspects is maybe they are transcribed and there is base pairing involved, and so we do see that some of them are transcribed. But there is a challenge that many people have found such sequences. In the fish, if it is this big, as you go more complex organisms like frogs or birds, the humans, they become bigger and bigger. So that means there is enrichment of something which is useful, but we don't know why it is. So it's as simple as that, but we don't know what their functions are. Many of these things one has to do experiment and figure out. They are very challenging to do. If we knock out, the cells don't survive.
Okay.
Then how to study it? It is challenging thing. Yeah.
Sir, I have a couple of more questions.
1 minute.
Can I finish it, sir? So, this is regarding the mRNA therapeutics, actually.
Yes.
I'm a little uncomfortable because this is synthetic mRNA or in vitro transcribed.
It's in vitro transcribed.
Because there you'll have some truncated transcripts also.
Yeah.
Achieving, when the length becomes bigger,
Yeah.
you'll have more challenges in getting full length transcript.
Yeah.
That is going to be more harmful than. So, how do you address
It's
these
a very
challenges?
important question, and that is what is the setting up the platform. So the way we do is we make a plasmid in which we get the gene synthesized, make a clone, and do transcription with T7 polymerase. And we, it's not only that, we have to introduce pseudouridine, and we do capping 3' and 5' so it doesn't get degraded. And the key thing is then after do HPLC purification and take only the full length one.
Okay.
And that is you got the most important point. Once you've got that, then the process is very simple after that.
That
Yeah.
cost is also going to be. I
It's
thought
very
in vitro
cheap.
transcription is very costly, actually.
You think it is costly, it is not costly.
It's
We
not
make
costly.
everything in the lab.
Okay,
T7 RNA. It's just, I'm saying the one thing will, and of course scale is important. We make in grams now. So, and we are not spending hundreds of crores. Our budget is small.
The last question is on the tissue targetability of, for instance, not every rare genetic disease is going to have same tissue problems.
Yes.
So when you put these liposomal or I don't know LNP with mRNA, can you surface modify them to target a
Yeah.
specific tissue
Yeah.
instead of?
Yeah, that is a $1 million question. And this is something which, one aspect which I didn't speak at all is what we are doing. Because like you said rightly, everything is not in the liver. Actually, if you inject, it should go to brain or muscle or something. And there are ways, in the LNP when you are making, because you are making in the lab, you can introduce a tag which will drag it to the tissue. But this is what is happening, that's where big money is. If you make one, you make a patent and you become rich.
Sir,
But it is possible, people are doing it. We are also seeing some results. Yeah, please go ahead.
good afternoon. You may recollect, under your leadership, CSIR has launched disinfection systems during COVID, which was validated in your lab. As per the US FDA guidelines for 4 pathogens, and now you are, all these pathogens were valid in liquid state. And all those 4 pathogens for US FDA luckily was food pathogens. So now if you see recently, all the pathogens which is coming in sewage, fecal sewage, the Melinda Gates plants have been set for septage in Bangalore. Spain has scaled up these systems, ozone and UV, and they have showed that antimicrobial resistant genes, all pesticides, all types of pathogens, all 14 microbes which comes in drinking water is treatable. And Spain has shown 113 all the emerging micropollutants. And one of the Tata Trust, I don't remember, which is near Bididi, they have visited us. But in India, after surveillance, we have not adapted these systems at Bangalore. Because we have made a case in Karnataka Pollution Control Board. What is it in your hand? Simple system to scale it up. And US FDA has given guidelines. And recently US FDA has issued a guideline for water use in the food industry, where dual disinfection, Pseudomonas is coming in Bangalore in food industries. So when we have some simple solutions, and we were leaders during COVID to scale UV, ozones for COVID, validated in CCMB, but we don't take it. We have written proposals to CSIR, but what is your view, sir? These are simple solutions, because in Bangalore, all the apartment kids are playing in the garden where treated sewage is applied. Nowhere in the world this type of exposure is this to young kids in Bangalore. Thank you, sir.
It's very pertinent question and very challenging thing. And so few general comments that we as a country, we are very good in working in emergency mode. but when the emergency goes away, we relax. Then we don't plan. We don't plan anything. That is problem in most democracies, because the lifetime or runway for any government is maximum 5 years. 1 year they are trying to figure out their coordinates. Last year they are worried about next election, and remaining 2-3 years for something else. So we don't have a long-term planning of that is a very basic problem. In fact, it's not only India problem. It's anywhere there is democracy is the same problem, because after 5 years these guys worried about his survival, not worried about your infection. So, but there is no easy solution, because lack of democracy is, then we all will be complaining. So here I think the scientists have to work. That is where work in little bit insulated from the government fevers that come once in 5 years, and now in our country comes every year. Somewhere some election is happening is another problem. But we have to get insulated, and that is why private funding or philanthropy funding is very, very important, because that doesn't depend on the government. We don't care about the gem. We don't care about, not that we buy anything more expensive than you buy. So, but we don't have to worry about, because there is some certain kinds of accountability that comes. And similarly, what our country misses a lot is private funding for the long-term research goals. They will fund you for 6 months to develop this product, or 1 year to develop this, or CSR money something, because they are forced to give, they will give if you put their name somewhere, but not a strategic long-term funding. And that is one aspect which we are trying to do with few other organizations. We want to showcase them TICS as an example that if you put money, philanthropic money in science, it solves societal problems. So instead of giving food to hungry people, you can solve the hunger problem forever. So, but that is our responsibility, and then scientists have to, instead of complaining, we have to take responsibility what we can do. Like say GM is problem in this country, right? GM is a big solution, good solution, and actually we are eating GM food, which is coming from the Bangladesh or America or wherever. But we are not allowed to produce, even if the government has agreed. Everybody, GAC has approved GM mustard, but still Supreme Court people are entertaining. I will, the people are deliberately or with certain motives or with their genuine concern that GM is a monster, but we have failed in convincing people what is safe, what is not safe. So scientists should take responsibility. We also don't, we think that why I should go, rather I sit and write a review or go for a meeting. So we have to take responsibility, and that is something which has been lacking. Like each institution, now it's becoming little bit, but in the wrong direction. Like all institutes are asked to have social media, Facebook, X and all that. But that is again, we published that paper, we attended this poster, we attended this meeting. We should spread the message of science, right? That what is right, what is wrong. And if an institution like CFTRI says something about food, people will listen. So I think we should contribute from our side and slowly bring the trust of the people in us. Now the water problem that you are saying is very, very important thing, and that should be done. Like microbes, outside you can kill them, but inside we cannot drink Chlorox and kill the thing, but outside we can treat and handle things. In fact, we are working with BBMP, we are taking inlet and outlet and testing for various drugs, because in Bangalore, lots of this water goes for vegetable and crop growing and comes to our kitchen. So we are trying to see that which treatment is working well, and we are working with other organizations like Biome and all, who are working very hard on rejuvenating lakes. So it's a big problem, and I think government should do, but I think people, if we give importance, government also will give importance. So more awareness and all has to come, and I hope things are, when we are realizing, as I said, now we are seeing the emergencies, so we are suddenly responding to few things, making cleaning the lakes, and some lakes have been rejuvenated, and that's showing very good example there. I'm very happy to see the lake that you guys have made. So these are very good examples, they should be showcased.
I would like to congratulate you for excellent seminar, and I would like to say that you are taking India into global level with respect to genome technologies. With respect to the agriculture, what exactly the varieties which we are mentioning about the low glycemic index, are they targeting the high amount of amylose which is responsible, is the one which gives us more resistant starch when we cook the food, rather than amylopectin, which are the branched polymer. So that is the thing which I'm feeling, whether amylose is getting targeted when they are having this type of agricultural based technologies. That is the first thing what I would like to ask.
So what we are doing at the GI part, we are doing a mutational breeding, so which is using a standard genetic approach. Without knowing what is happening, you mutate and pick what is your liking, where the glycemic index is lower. Once we get those things, then we can go back and sequence and see what has changed, so that later we can do it in more precise manner. But we are not quite there yet.
Yeah, with respect to the health, we are mentioning most of the things are translation only, but what are the post-translation modifications which are going to have an impact with respect to these things that one has to address?
It's very important thing, so one has to look into those. We haven't seen that.
Okay. Yeah. Thank you very much.
Thank you, sir, for sharing your wisdom and knowledge in genome technologies and its expansion in crop improvement, food sciences, plant proteins, mRNA technologies, in evolution for biomedical sciences, and also the environmental sciences. Thank you, sir. Now, may I request our director to come over to the stage? So please be seated. Now may I request our director, Dr. Sridevi Annapurna Singh, for the presidential remarks.
Good afternoon to all. I think we all agree that it was a magical 1 hour that we spent listening. I was very curious when I saw the title, and the first slide said Science, Society, and how he would be linking it up in a very short time. But I was fascinated, even the first slide showed how the DNA is long enough to circle the sun 60 times and is still packed in such a small cell. And here we are spending a year trying to get one equipment from indenting to, clearing the bills. And we should learn from ourselves probably how to be more efficient. But having said that, I think it was a wonderful, wonderful talk. Thank you so much for accepting our invitation and showing us the possibilities. We live in very exciting times, I must say, from the science point of view, with a lot of things to look forward to. And the concept of One Health, I think food plays a very, very important role, because that is the agriculture and its allied industries have resulted in clearing of lot of forests, using of lot of fresh water. The soil health is affected because of this. And a lot of food gets wasted also. Nearly 30% of the food that we grow is wasted. So somewhere here I do find relevance of our institute, where we have to ensure that we are using lesser energy, greener technologies for the post-harvest processing of foods, which will also help to increase the bioavailability as well as the digestibility. That would add value to whatever is done from the agricultural viewpoint of crop breeding or mutation breeding and so on. And a very interesting fact I just wanted to bring to your notice, that recently during the International Year of Millets, we did have Millet Mission program under CSIR, and CFTRI was the nodal laboratory for that. It is going to conclude in March 2025. And we did test profile nutritionally a large number of millets, and some of them were biofortified. So we saw the iron content was something like 9 milligrams and so on, but the phytates also went up 3 times more. So I'm not very certain that we have gone smarter than nature. Somewhere the plant is also trying to protect its seed by ensuring that there is a counter. So a little more holistically, we would probably have to look at it if we really want the iron to be bioavailable and to be digestible on ingestion. So as we go to personalized nutrition, we are already into that. Each one of us within the family are also eating different foods. So I think today's lecture was extremely concentrated and focused towards that. We have been trying to make foods for rare disease, and we are taking baby steps. We are still not, but we are looking at all of that. And one incentive for all of this probably was the numbers that you showed, the market size. It's so many billions and billions of dollars. Unfortunately, in our country, if you are poor, you have very little access to health care, especially if the disease is kind of rare. So I think you have shown us the path where CFTRI should be working in the future also through your talk, and thank you once again very much. Thank you.
Thank you, ma'am, for your remarks. Now, may I invite Dr. Girdhar P. onto the dais. I request our esteemed director to kindly felicitate our chief guest, Dr. Rakesh Mishra ji. Now, may I request Dr. Asha Martin, Senior Principal Scientist, Department of Biochemistry, to give the vote of thanks.
Jubilee lecture series, I deem it a great honor to propose the vote of thanks. Ladies and gentlemen, this lecture series is being organized to commemorate 75 years, an important milestone in the history of our institute. We are extremely privileged to have as a speaker Dr. Rakesh Mishra, Director, TIGS, whose remarkable contributions to genome organization, chromatin structure, and epigenetic regulation has left an indelible mark on both Indian and global science. Sir, we are so honored that you could take the time to be part of this prestigious lecture series. Thank you for sharing your valuable insights on various genome technologies and highlighting their role in ensuring nutritional security, environmental surveillance, and healthcare for all. I would like to express my gratitude to our beloved director, Dr. Sridevi Annapurna Singh, for a gracious presence and for giving the presidential remarks. On behalf of the organizing committee, I thank our director, ma'am, for her unfaltering support. She's the backbone of our institution and the guiding force for this event. Thank you, ma'am, for all your support and encouragement. I'm pleased to extend my warmest appreciation to everyone who has worked on this event. We are grateful to Dr. P. Giridhar for his overall guidance, Dr. Purnima for taking the lead in organizing this event, and to all the organizing committee members for their excellent cooperation in conducting the lecture series. I would be failing in my duty if I don't thank all my dear colleagues from PMC, IT and CS, INP, Agri-Horticulture, EMM team who have helped us in making this event possible. My deep sense of thanks and appreciation goes to all who are attending this event online, staff and students of CFTRI present here, and our esteemed retirees for attending the lecture series and making it a great success. At the end, I thank you all so very much for being with us today and look forward to your gracious presence in the upcoming lectures as well. We have some exciting lectures and eminent speakers lined up for this prestigious lecture series. Have a wonderful day ahead. Thank you.
Thank you, ma'am. Now I request all of you to kindly stand up for the national anthem.
जनगण मन अधिनायक जय हे भारत भाग्य विधाता पंजाब सिंध गुजरात मराठा द्राविड उत्कल गंगा विंध्य हिमाचल यमुना गंगा उच्चल जलधि तरंगा तव शुभ नामे जागे तव शुभ आशीष मांगे गाहे तव जय गाथा जन गण मंगलदायक जय हे भारत भाग्य विधाता जय हे जय हे जय हे जय 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 हे
Thank you.